Right. So let's continue talking about the difference between normal cells and cancerous cells. So I just want to reemphasize the cell structure here. Plasma membrane is a surrounding structure around all of our cells. It's made up of phospholipids, these fats. The nucleus is found in all of our cells, and this is what contains our DNA. Now, when the cell is sitting in this part of the cell cycle, where it's just hanging out, so to speak, the DNA is called chromatin. As the cell goes through the division process of the cell cycle, that chromatin changes into what we call the chromosomes. So most of the time we're familiar with the term chromosome. We have 46 chromosomes, or this is on this piece of DNA or gene is found on a certain chromosome. It's all the same stuff. It's just the organizational state of the information. I like to think about chromatin like a big pile of spaghetti noodles. It's just this big jumbled mass of stuff. That's chromatin. And we'll walk through the cell cycle and see how does the cell go through each step of the cycle, what happens to the genetics and the DNA. So, all right. So anyway, what we were talking about before, basic components, and then your cytoplasm, of course. Let's not forget about that. Cytoplasm is just kind of that fluid, liquidy space. But there's physical differences when we look at a cancer cell versus a normal cell. And this is one way we can identify if someone has cancer. As I mentioned before, you take that cell out. You take a little cutting of cells out, or a sample of the tissue, and then let's analyze it. Let's put it under a microscope and actually look at the shape. That's the starting point to then say, okay, are these cells abnormal? So if it's caught early, as I will continue to stress, maybe it's a simple procedure of, well, let me remove all of the cells that potentially have cancer in them. All right, so very poor drawing of a nose here. And imagine you have a couple cancer cells, or you have a little tumor, a little mole, a little growth, a little weird spot, something that doesn't look right. When the biopsy is taken, physician will come in and, okay, let's cut that out. And now let's take a look at those cells. Are they abnormal? Are they cancerous cells? If identified as yes, then they want to go back and they want to remove all of those cells. So this is why, all right, we got to try to get this whole group of cells out, eliminate those, and leave none behind. Hypothetically, if they missed a couple of these cells are just going to continue to go through the cell cycle and your tumor is going to grow. So we got to get rid of them all is the goal. Eliminate all of those cancerous cells. But structurally, looking at these cells at the cellular level, there are differences. So here's breast cancer versus a normal breast cell. On the cells, that plasma membrane, are these little receptors. So the receptors here, let me put the arrow here. These are little protein structures that identify your cell. Hey, I'm a cell for you. I have a unique structure, a little shape. I can attach to other cells. I can allow certain molecules to come in because of this receptor. The protein receptors play a huge role in how our body works. If you get a viral infection, the virus attaches to your receptors. And that's why some people are less likely to get infected than others. It's based on protein receptors. But when we compare a breast cancer cell versus a normal breast cell, the number of receptors is significantly different here. The one on the right is a breast cancer cell. And look at the number of protein receptors versus the one on the left. So this is connected to the HER2 protein. This protein, human epidermal growth factor, is the protein that is attached to the cell membrane. Cancerous cells, breast cancer cells, have tons of these receptors. That's not a good thing. That's what tells the cell when to divide. And what we're seeing statistically, about 1 in every 5, oh, that's not supposed to cover it. 1 in every 5 cases of breast cancer has a mutation in your DNA that causes the cells to make a whole bunch of these proteins. These excessive proteins are what mess up the cell cycle. Uncontrolled cell division because you have so many extra proteins there because of a gene. So there's the whole connection, or not. Not that that's it. It's not that simple, but there's a step in understanding the connection. So you have a gene, a piece of DNA, that tells your cell, make a bunch of these. You make a whole bunch of these, and now that cell divides uncontrollably and produces breast cancer. That's one way. There's lots of different ways that this can happen, but that's just one example of how cancer can form. This is your genetics. You can't change your gene at this point. Do you know if you have these genes? And this is where I'm going to encourage everybody to think about their family. Look at their family tree. Ladies, ask your mothers, ask your grandmothers, your genetic aunts. If they've married into the family, you still want to have concern for your family members, but their genetics don't influence yours. It's direct genetic lineage. Your mother, your grandmother, aunts, sisters, cousins, every female who has a genetic connection to you, ideally you get this information to then start looking at what are the odds you inherited that gene. And if there's concern about having inherited that gene, you bring this up with your physician and say, I think I may be at a high risk for breast cancer because my mother had it, my aunt had it, my grandmother. Unfortunately, the odds are you may have inherited that gene. Now we need to be more aware. Perhaps mammograms earlier in life to detect if you are developing a tumor instead of ignoring it, not worrying about it. So I want you guys to start really thinking about the bigger implications of all this. If I understand I have a high risk for this, can I get tested earlier? Will my health insurance pay for this? Will it cover testing earlier because I have a potential genetic connection to something like this? This is why this is a big, big issue. It ripples out and impacts everything in our lives, from our personal health to our paycheck, to insurance, to social programs. There's no part of society that's not touched by these issues. That's why they're so relevant to all of us, because we connect to it. So, all right, here's one known as Hodgkin's lymphoma or Hodgkin lymphoma. We look at the cells, normal lymphocyte, and then the cells that change are now, they're referred to as the Reed-Sternberg cell. That's the big one in the center here, this one here. And it's big, it's weird. It's got 3 nucleus. You shouldn't have 3 inside one cell. Each cell should only have 1 nucleus. This cell has 3? That's not right. That's an indication something's wrong and there's a problem. So, biopsy, pull the cells out. Can we identify if they're there? Then can we now treat it? Leukemia, blood cells, cancer of the blood. Now, red blood cells, typically we're not worried about those. Those are those little things that look like jelly donuts here, the red blood cells. It's the white blood cell. When we talk about leukemia, we're referencing white blood cells. Are they normal? Are they abnormal? And you can see over here, this is healthy blood, the shape, the general features of the normal white blood cells. We come over here, and we start looking at shapes a little different, but how many of these we have? There's a whole bunch of these. There's lots of these white blood cells in leukemia. So your body is cranking out tons and tons of extra white blood cells, but they're not functional. White blood cells protect the body. That's their job. You get sick, you have a bacterial infection, white blood cells go to town. You cut your leg, white blood cells go to defend the injury. That's their job. They're the defenders. When somebody has leukemia, there's a whole bunch of extra white blood cells produced, but they tend not to do their job. And now you're thrown out of homeostasis. Thyroid cancer. Normal thyroid follicle. It's a ring of cells. Oh, we got something growing here that shouldn't be growing there. Those cells continue to grow, continue to grow, continue to grow because, hey, they're not going to stop their cancer. And that thyroid follicle is now covered and literally drowned out and overcome by a mass of useless cancer cells. So the thyroid's not able to produce the hormones it should. It's not functioning correctly. We're out of homeostasis. That's what this stuff does, is it throws the body off. So I want you guys to think about a vehicle you ride in, whether it's you have a car, a motorcycle, a bicycle, you jump on the bus. Imagine if I bent one of the wheels, twisted the wheel so it didn't roll smoothly. Think about how that would impact the entire vehicle. The other tires are going to get messed up. The frame's going to start to get twisted and warped and bounced. Everything inside the car is going to bounce around and rattle a bunch. Now all of a sudden your mirror falls off and your radio doesn't work because it's been bounced around so much. That's what we're seeing happen here, is it's that cascade effect of taking a system out of balance and how that ripples through all these other systems. So huge, huge problems. And that's why we want to try to identify this stuff as early as possible. Get it identified and let's get it eliminated. Let's destroy those cells. So, all right. So cancer, as I mentioned before, I'm going to put this back up again because hopefully you're getting the hint here what parts are critical to know and stressed. Cancer, the cells don't follow the rules. Now, one reason why they may not follow the rules is you have a gene, a genetic code that causes them to act differently. It's a different piece of instruction that tells the cell, do something differently. You may have a gene that says, it's acting differently. We're not going to stop it. Keep going. That's okay. Or there could be an environmental factor that changes the gene and then causes the cell to act differently. So there's lots of different ways that these cells can not follow the rules or what can contribute to them not following the rules. And the goal is to identify how to keep them from changing and hopefully make them follow the rules. So we'll look at that in the next part.